At the end of this topic, you will be able to define heredity, inheritance, traits, list out the characters chosen by Mendel on pea plant, explain the monohybrid cross with checkerboard, distinguish allele from allelomorph, detail the variation with its types and its role in evolution, compile the life history and research interest of Gregor Johann Mendel. The genetic transmission of characteristics from parents to offspring is called heredity. Variation is the change in the characteristics between the individuals or between the groups of population. Inheritance in biology is the process in which an offspring possesses the determined characteristics of its parent. The morphological, anatomical, physiological, and reproductive characteristics passed through inheritance are called as traits. The basic rule of inheritance is both the parents are equally contributing in passing a trait to their progeny. Parents are passing their genetic traits via DNA to their offspring. Gregor Johann Mendel worked out scientifically for the first time on heredity and variation in pea plant, Pisum sativum, which he had cultivated in his monastery garden. Mendel was curious to get the results of crossing various traits. He chose 7 contrasting characters for his study and worked out continuously on that. The 7 contrasting characters were as follows. Shape of the seed with 2 contrasting characters such as round and wrinkled. Color of the seed with 2 contrasting characters such as yellow and green. Color of the flower with 2 contrasting characters such as violet and white. Pod shape of the fruit with 2 contrasting characters such as full and constricted pods. Pod color of the fruit with 2 contrasting characters such as green and yellow. The position of the flower in a plant with 2 contrasting characters such as axillary and terminal. The height of the plant with 2 contrasting characters such as tall and dwarf. Mendel selected the Pisum sativum plant with 2 different heights for his observation. Pea plants generally produce seeds by self-pollination, and he noted that the tall plants produced always tall plants generation after generation and dwarf plants produced always dwarf plants generation after generation under natural condition. Mendel termed them as wild types or pure breeding varieties. Mendel crossed the pure breeding tall and dwarf variety of pea plants. He crossed a tall plant with a dwarf plant for subsequent generations and calculated the percentage of tallness and dwarfness. When a pure breeding tall plant was crossed with a pure breeding dwarf plant, all plants were tall in the first filial generation, F1, and dwarf character was not appearing at all. He self-crossed the F1 generation to check whether dwarf characters will express in the next generation. As he expected, dwarf character was expressed in F2 generation with the ratio of 3 tall plants and 1 dwarf plant. At the end of the experiment, he concluded that dwarf character was suppressed in the F1 generation but expressed in the subsequent generation with meager percentage. He termed phenotype as an expression of morphological characters as tall or dwarf, red or white flower, and genotype as the expression of the gene or chromosomal makeup of an individual for a particular trait. The genotype of a character is determined by factors later called as genes. Genes are the main physical basis for inheritance of characters. The contrasting expressions of the single gene are called alleles. Contrasting pair of alleles forms an allelomorph. Tall and dwarf plant and white and violet colored flowers are alleles. Height of the plant and color of the flower is an allelomorph. Organisms differ or vary in expressing their phenotype is the key factor for variation. No 2 individuals are identical to each other and they are unique in their own ways. The difference in the characteristics among the individuals is called variation. Variation may be within the same species called as intraspecific variation or between different species called as interspecific variation or among the different genera called as intergeneric variation. Asexual reproduction produces more or less resembling offsprings which result in minor variations. Sexual reproduction brings about remarkable, significant and visible variations. Somatic variations are pertained to body cells and are not inherited. Germinal variations are pertained to gametes and are inheritable. Germinal variations pave the way for speciation and evolution. Variation is the raw material for evolution. Variation makes animals to adapt themselves to the changing environment. It brings out the best in animals to face the struggle for their existence. Individuality of an organism is well maintained. Variation is a keystone for heredity, otherwise all individuals of a race would be identical in all aspects. Gregor Johann Mendel lived between 1822 and 1884. Mendel was an Austrian Augustinian monk by profession. He was educated in a monastery and went on to study science and mathematics at the University of Vienna. He failed in teacher training examination, but it did not suppress his thirst on experimental research. He turned to monastery and became a pastor, but adoring gardening and research. He started growing peas on his monastery garden. Many others had studied the inheritance of traits in peas and other organisms earlier, but Mendel blended his knowledge of science and maths and was the first one to explore the scientific method of keeping the count of an individual's exhibiting a particular trait in each generation. This helped him to frame the laws of inheritance and gave him the title Father of Genetics posthumously. In this topic, you have learnt heredity is the genetic transmission of characteristics from parents to offspring. In inheritance, offspring possesses the determined traits of its parents. Gregor Johann Mendel, father of genetics, worked out scientifically on pea plants on his monastery garden, selected 7 different alleles, observed generation after generations, and concluded his genetics work mathematically for the first time. In his monohybrid cross experiment, he selected tall and dwarf plant and observed F1 and F2 generations and concluded with the phenotypic ratio of tall and dwarf as 3:1 respectively. He termed gene as factors and its expressions as genotypes. He mentioned alleles are the contrasting expressions of the single gene and genes are the physical basis of inheritance too. Every organism differs from the other of their own species, which is called as variation. Asexual reproduction shows minor variations, whereas sexual reproduction expresses significant variation. Variation may be somatic or germinal, and it is the raw material and keystone for evolution. At the end of this topic, you will be able to define evolution, explain how new species are formed naturally, list out the hierarchy of human evolution from hominids to modern Homo sapiens, illustrate the diversity of organisms with evolutionary tree, brief the use and disuse theory of Lamarck, compile the voyage and the researches of Charles Darwin. Evolution is a gradual process in which something changes into a different and more complex form from the pre-existing simpler forms. It is rather a slow process which would take millions and millions of years. Generally, evolution is studied or presumed by fossil evidences. Evolution brings out diversity in organism. It is basically influenced by environmental selection. Speciation is formation of new species from a subpopulation of a species which is geographically or reproductively isolated over a long period of time. When two populations or species are isolated by geographical, anatomical, physiological or reproductive barriers for a long time, there is a chance for them to have a change in their gene flow. Gene flow ultimately leads to speciation. Gene flow in a community or population may bring out the genetic drift. Genetic drift along with gene flow imposed by isolation mechanism is a good cause and agent for speciation. Hairy-bodied gorilla and chimpanzees like hominids existed in Africa some 15 million years ago. Around 3 to 4 million years ago, men like hominids walked into eastern Africa. Fossil evidences say that hominids used stones for hunting and were mostly eating fruits. They were short with a height of not more than 4 feet, but walked upright in the grasslands of East Africa, looked men-like and called as Homo habilis. There was a long gap between in human evolution or lack of adequate fossil evidences as the next stage of human evolution came to exist 1.5 million years ago, and they were called as Homo erectus, who were meat-eaters. 1 million years ago, the Neanderthal man lived in East and Central Asia. They protected themselves by hiding and buried their dead companions. Archaic Homo sapiens arose in South Africa and moved across continents. They developed a distinct race during the Ice Age. The modern Homo sapiens arose between 75,000 to 10,000 years ago. This was evident by the prehistoric cave which was developed about 18,000 years ago. Around 10,000 years back, agriculture came to practice where human settlements started. Evolutionary tree is used to understand the evolution, variation and relationship between various species and other entities. Its branched diagrams typically looks like trees. This is drawn based on the similarities and differences in physical and genetic characters of biological species. Jean-Baptiste Lamarck lived between 1744 to 1829, postulated the use and disuse theory. According to Lamarck, the organ which was in use frequently for generations over a long period develops well in the subsequent generations, and the less used or disused organ or parts of a body diminishes or degenerates in the subsequent generations. Lamarck cited the development of long neck of giraffe as an example for use and disuse theory. Initially, giraffes had a short neck, but they were forced to stretch their neck and leg to reach the leaves of tall trees. This action was continuing over a period of time, which resulted in long neck and legs in giraffe. Lamarck quoted that will or want for a character makes the organisms to grab it at a later time. Charles Darwin, the father of evolution, set out on a voyage when he was just 22 years old. He was sailing for 5 years and visited South America and the Galapagos Islands. After this voyage, he had never visited any shores but stayed in his home country, England, and conducted various researches pertaining to evolution. After the elaborate research he formulated, the hypothesis that natural selection is the root cause for evolution. Unfortunately, he did not have an idea or knowledge about the mechanism of variation seen in the species. Had he been highlighted the ideas of Mendel and his experiments on variation and genetics, he would have contributed more to the evolutionary thesis. Darwin was not only an evolutionary biologist but an accomplished naturalist and conducted notable research on earthworms' role in soil fertility. In this topic, you have learnt evolution is a slow, gradual process in which something changes into complex form from the pre-existing simpler forms. Evolution brings diversity in organisms. Formation of new species from a subpopulation of a species is called speciation. A barrier existing for a long time brings out gene flow, genetic drift, and speciation. Human evolution started from hairy-bodied gorilla and chimpanzees like hominids 15 million years ago, to men like hominids 3 to 4 million years ago, to Homo habilis to Homo erectus 1.5 million years ago, to the Neanderthal man 1 million years ago, to archaic Homo sapiens to the modern Homo sapiens 75,000 to 10,000 years ago. Humans settled around 10,000 years back when agriculture came to practice. Evolutionary tree is drawn used to understand the evolution, variation and relationship between various species and other entities. Jean-Baptiste Lamarck lived between 1744 to 1829 and postulated the use and disuse theory, quoting the example of the giraffe's neck. Charles Darwin, the father of evolution, set out on a voyage to South America and the Galapagos Islands and spent 5 years, came back to England, researched extensively and formulated the theory of natural selection. At the end of this topic, you will be able to define what genetic engineering is, name the enzymes involved in genetic engineering, specify the uses of genetic engineering, list out the scope and uses of biotechnology, define what clone is, explain the techniques of cloning, discriminate the natural clone from artificial clones, describe the stem cell culture and its uses, distinguish the embryonic stem cell from somatic stem cell, tell the uses of biosensors and biochips, summarize the significance of gene therapy, compare the somatic gene therapy and germline gene therapy. Recent advancements in genetics, molecular biology and biochemistry has opened a new branch of science called genetic engineering. Genetic engineering is the manipulation of DNA of a living organism by removing or adding or repairing the part of it. It is also known as recombinant DNA technology or rDNA technology. Genetic engineering helps in understanding the gene structure and function through basic research. It helps in the production of large quantities of insulin, interferon, antiviral proteins produced by virus-infected cells, human growth hormones, proteins, polypeptides and vaccines, etc. Its uses in agriculture are vast as it helps in transferring the nitrogen-fixing gene in many plants which enhance the productivity. There are two basic enzymes in genetic engineering called restriction endonuclease and ligase. Genetic engineering has developed after the discovery of these two enzymes. Restriction endonucleases or restriction enzymes act as scissors which cut the DNA in specific sites, so they are called as molecular scissors. Ligases act like a paste which helps to join the broken DNA fragments. Biotechnology is mainly used to exploit the organisms or biological processes for profit in medicine, agriculture, animal husbandry, and environmental cleaning. Biotechnology is also used in several industries such as brewing industry, enzyme technology, vaccine production, etc. There are several applications of biotechnology such as metabolic extraction, waste treatments, production of medicines, vaccines, etc., artificial flavor and food production, production of organic acids such as acetic acid, citric acid, etc., extraction of minerals, releasing transgenic plants and animals, biogas production. Fermentation technology is the major area of biotechnology which is used in the production of alcoholic beverages like beer, wine, etc. Enzymes are biocatalysts that speed up reaction in cells. They are isolated from the cell and used for industrially important reactions. Improvised enzymes are more efficient than inorganic catalysts. Antibiotics are the substances produced by some microbes which help to increase our immune system, and they are produced in large quantities via biotechnology. Acetic acid is one of the most important acids in vinegar production, which is produced expansively in industries with the help of biotechnological applications. Vitamins are the chemical compounds present in minute quantities in natural foodstuffs but very essential for our survival. They are made in large quantities using biotechnology. Vaccines induce immunity against specific diseases and act as antigens and stimulate the antibody production. Steroids are derived lipids. Prednisolone is a drug which contains cholesterol which is produced from fungus rhizopus. Monoclonal antibodies are the antibodies produced by clone cells and used to treat cancer cells. Biotechnology plays a vital role in microbial production, and both biotechnology and microbiology are interdependent. The microbiology products like vaccine, antibiotics, enzymes, and vitamins are produced through biotechnology. Vitamin B12 is synthetically made and helpful to cure pernicious anemia. The mass production of insulin in biotechnology is a boon for diabetic patients. Biochemically significant enzymes such as amylase is technically produced from bacterial amyloproteins. Cloning is a technique of producing morphologically and genetically identical organisms in groups. The clone is an organism produced from a single parent by asexual methods but having diploid chromosomes. A clone is a Xerox copy of a single parent, and it can be done only with living species. If the cloning technique is properly applied in veterinary science, valuable animals could be made from desirable adult cells. There are two types of clones, namely natural clones and induced clones. Identical twins come under natural clones. The induced or artificial clones are developed by transferring nucleus into the host cell. Stem cell culture is the fascinating branch of applied embryology. The stem cells means unspecialized mass of cells which derive either from plant or from animal. The two important striking feature of a stem cell is cell should be unspecialized but should have the potentiality of growing and multiplying mitotically into countless number of same type of cells. They must have the capacity to imbibe the specific function of the tissue which it has been introduced. For example, they may be induced to become a cardiac muscle or beta cells of pancreas for insulin production, etc. There are two kinds of stem cells, namely embryonic stem cell and adult or somatic stem cell. The embryonic stem cells are made by in vitro fertilization. Zygote forms a hollow blastula with repeated mitotic division where the destination of cell was not determined. The inner mass of undifferentiated cells of blastula is isolated and used as embryonic stem cells. There are many well-differentiated tissues like epithelial, vascular, muscular, etc. in the body of higher animals and human beings. There will be some undifferentiated cells seen in these tissues which can be considered as stem cells. They can be isolated and grown artificially as a destined cell, and the mechanism of growth is similar to that of embryonic stem cell. Bone marrow, embryos, amniotic fluid, and umbilical cord are the best sources for somatic stem cells. Biosensor is a device carrying immobilized layer of biological materials like enzyme, antibody, hormone, nucleic acid, organelles, etc. having contact with a sensor. The sensor in the device converts biological signals into an electrical signal. Biosensors are widely used in medicines and industries. Blood glucose level can be detected quickly using biosensors. Toxin production in the body due to infection can be detected. The amount of pollutant in drinking water can be monitored. The quantity of food such as odor, freshness, and taste can be measured. Biochips are microchips which are developed by the techniques of biotechnology. Biological computers with biochips will be developed in future. Biochips are used in defense, medicine, etc. Using the principles of biotechnology or genetic engineering, the defective gene can be replaced or repaired. For example, the disease diabetics caused by the degeneration of beta cells in pancreas can be rectified by altering the insulin-controlling gene. Gene therapy is the bright way to cure genetic diseases and incurable acquired diseases like AIDS, etc. It can also be used to treat the defects in somatic cells and gammatic cells. There are two types of gene therapy, namely somatic gene therapy and germ-line gene therapy. In somatic gene therapy, the genome of the recipient is changed and it is not inherited to the next generation. In germ-line gene therapy, the egg and sperm of the parents are changed so as to pass the changes to the next generation. In this topic, you have learnt genetic engineering or recombinant DNA technology is the manipulation of DNA of a living organism which helps to understand gene structure, production of insulin, interferon, etc. Biotechnology is used to exploit the organisms or biological processes for profit in medicine, agriculture, etc. and is widely used in brewing industry, enzyme technology, waste treatments, biogas production, etc. Cloning is a technique of producing morphologically and genetically identical organisms. The stem cells is an unspecialized mass of cells, should have the potentiality of multiplying mitotically into countless number of same type of cells and must have the capacity to imbibe the specific function of the tissue which it has been introduced. The embryonic stem cells are made by in vitro fertilization from the inner mass of cells from the blastula. Somatic stem cells are taken from epithelial, vascular, muscular tissues, but bone marrow, embryos, amniotic fluid and umbilical cord are the best sources for somatic stem cells. Biosensor and biochips are biotechnology products used in medicine, defense, pollution boards, etc. Gene therapy is used to treat incurable diseases, genetic disorders, etc. and there are two types, namely somatic gene therapy and germ-line gene therapy.